Hello, welcome to Study Hub Pro. So, today we're diving into a really cool topic, transduction. It's one of the most fascinating ways bacteria share their genetic information. I want you to imagine, for a second, viruses acting like these tiny, almost accidental delivery drones, just shuttling DNA from one bacterium to another. So, how does this incredible process actually work? Well, let's get into it. Okay, here's a quick look at what we're going to cover. First, we'll talk about the viruses themselves, the genetic hijackers. Then we'll look at their life cycles. From there, we'll get into the two main types of transduction, generalized and specialized. And we'll wrap up by looking at why all of this really matters. Sounds good? Let's jump in. All right, first up, let's meet the key players in this whole story, the viruses. To really get a grip on transduction, we've got to understand these tiny little agents that are making it all happen. So what exactly is a virus, anyway? Well, it's kind of in this weird gray area. It's not really alive. The best way to think about it is like a little package of genetic instructions all wrapped in a protein shell. It can't do anything on its own. To reproduce, it has to pull off what's basically a molecular hijack. It invades a living cell and takes over its machinery to churn out more copies of itself. Now for what we're talking about today, we need to focus on one specific type of virus. And that is the bacteriophage, or just phage for short. I mean, look at this thing. It looks like something straight out of science fiction, like a tiny lunar lander. And its one and only mission? To infect bacteria. Okay, so before we go any deeper, it helps to see where transduction fits in the grand scheme of things. See, bacteria are really good at sharing genes with each other, and this whole process is called horizontal gene transfer. They've got a few tricks up their sleeves. There's transformation, where they just grab DNA floating around them. There's conjugation, which is like a direct cell-to-cell transfer. And then there's our topic for today, transduction, the one that's all mediated by viruses. So we have our phage. How does it actually infect a bacterium? Well, it basically has two different playbooks it can run. These are its life cycles, the lytic pathway and the lysogenic pathway. And trust me, you really need to understand both of these because they are the key to telling the two types of transduction apart. First up, we've got the Lytic Cycle. This is the fast, aggressive, get in and get out approach. The phage lands on the host, injects its genome, and boom, the hijack is on. It immediately starts breaking down the host's own DNA and tells the cell, nope, you work for me now. The cell becomes a factory, replicating the phage's genome and building all the new virus parts. Once everything's assembled, the grand finale. The cell bursts open, it lyses, releasing a whole new army of viruses. And this image here gives you a great visual of that whole process. You can see it all happening, right? The phage attaches, injects its DNA, the cell turns into a factory for new phages, and then, pop, the cell is destroyed. It's a total hostile takeover from start to finish. But that's not the only way. There's a much sneakier option, the Lysogenic Cycle. Think of this one as the sleeper agent approach. Instead of an immediate takeover, the phage DNA does something really clever. It actually weaves itself into the host bacterium's own chromosome. And once it's in there, we call it a prophage. It just lies dormant, totally silent, getting copied right along with the bacterial DNA every single time the cell divides. Okay, so this is the absolute key takeaway here. The lytic cycle is what we call a virulent infection. It's active, it's destructive, it happens right away. The lysogenic cycle, on the other hand, is temperate. The virus is just dormant, hiding out as a prophage. It's waiting for some kind of stress signal, like, say, UV light, which tells it, okay, time to wake up. Then it will excise itself from the chromosome and kick off the lytic cycle. And I can't stress this enough. Understanding this difference is going to unlock everything else we're about to talk about. All right, now that we've got the lytic cycle down, we can talk about our first type of transduction. It's called generalized transduction, and it's basically an accidental byproduct of that fast and furious lytic invasion we just saw. And the key word to remember here is generalized. Why? Because any random piece of the bacterial DNA can get transferred. This all comes down to a simple mistake. Picture the scene. The cell is in total chaos, frantically assembling new virus parts. In the rush, the packaging machinery messes up. Instead of grabbing the viral genome, it accidentally stuffs a random fragment of the host's own chopped-up DNA into a new phage head. So let's walk through it. Step 1, a phage infects the donor cell, and the lytic cycle begins. The first order of business for the virus is to release enzymes that just shred the host's chromosome into a bunch of little pieces. And here's where the critical mistake happens. As all the new phages are being assembled, one of them gets packed with a piece of that bacterial DNA by mistake. The actual viral DNA for that particle gets left behind. So now you have a phage head that's full of host DNA. This faulty virus is what we call a transducing particle. On the outside, it looks and acts just like a normal phage, so it can go and infect a new bacterial cell. But here's the twist. When it injects its genetic material, it's not injecting viral DNA. It's injecting that piece of DNA from the original host. And if that new piece of DNA finds a home and integrates into the recipient's chromosome, boom, that's genetic exchange. That's generalized transduction.
Okay, that's one type down. Now let's look at the second kind, which, as you might have guessed, is tied to the lysogenic cycle. This is called specialized transduction.
So if generalized was all about random packaging, specialized is, well, much more specific. This all happens because of a different kind of mistake. It's an error that occurs when a prophage decides it's time to leave the host chromosome and kick off the lytic cycle. The key here is that when it makes a mistake cutting itself out, it can only grab and transfer the bacterial genes that are right next door to where it was integrated. Let's take a closer look at how this works. So here you can see the prophage, just sitting there in the host chromosome. Now, when it gets a signal to leave, the idea is to make a nice, clean cut. But sometimes it's a sloppy excision. The cut is imprecise, and when the prophage pops out, it accidentally takes a little piece of the adjacent bacterial DNA along for the ride. Now, the phage that results from this is actually defective because in taking some bacterial DNA, it had to leave some of its own DNA behind. So what you have now is a particle carrying this hybrid DNA molecule, part virus, part bacteria. When this phage goes on to infect a new cell, it injects that hybrid DNA. And that's how you get a very targeted, very specialized transfer of only those specific genes that were next to the prophage. Okay, this table right here is perfect for studying. Seriously. It breaks down all the key differences you need to know. Let's run through it real quick. Generalized transduction, that's the Lytic cycle. It transfers any gene, and it's all because of a random packaging error. The phage particle itself only has bacterial DNA. Now, specialized transduction, that comes from the Lysogenic cycle. It only transfers genes next to the prophage site, and the cause is a sloppy excision error. And importantly, that resulting phage has both bacterial and phage DNA. If you can remember this table, you're in great shape.
All right, so you might be thinking, this is cool microbiology, but why does it really matter? Well, the reason we study this in so much detail is because understanding transduction has given scientists an absolutely incredible toolkit for things like genetic engineering. I mean, we've basically figured out how to hijack the hijackers. We use this natural process for all sorts of amazing things, like mapping out entire bacterial genomes, figuring out which genes are located near each other, and of course, for genetic modification. But honestly, one of the most game-changing applications has to be in the field of gene therapy. And this is exactly what we're talking about when we say viral vectors in gene therapy. It's brilliant, really. Scientists take a virus, they strip out the parts that make you sick, and they replace them with a working, healthy copy of a human gene. The virus then becomes our delivery drone, carrying that therapeutic gene right into a patient's cells where it's needed to fight a genetic disease. So, I'm going to leave you with one final question to chew on, something that connects everything we've talked about to a huge real-world problem. We've just seen in detail how viruses can act as shuttles, moving genes between bacteria. So, think about one of the biggest threats we face in medicine today: antibiotic resistance. How do you think a process like transduction could be fueling the super fast spread of resistance genes throughout the bacterial world? See you in the next video.